it's 2 minutes to go. Anybody know Richard Pearson or Thomas Daly? Sorry, who are you? Sorry, you know them, right? Are they coming? Okay, so we've got everybody but Richard Pearson. Richard Pearson with 1 minute to go. Richard Pearson. We believe he's here. We've got to talk from him. Welcome everyone. It's 11 o'clock, so it's my job to start us on time. This is the third of 5 sessions on global change ecology during this conference, and this one's distribution, dynamics and models. And our first speaker is Yvonne Buckley from Trinity College, Dublin, and she's talking about predicting plant populations' response to global change. Yvonne.
Okay, so I'm going to talk about predicting plant population responses to global change. We need to be able to predict plant population performance in order to predict the extinction and persistence of unstudied species. So these are species for which we may have no or very limited information, and in locations where we may not have studied population performance previously. We need to be able to predict how species will respond to the conditions that they encounter when they're taken outside of their current range and put into their non-native ranges. And we need to know what's going to happen to populations under new environmental conditions, such as changes in climate and in particular. So what I'm going to talk about today is what we can use in order to make predictions about population performance. The first approach I'm going to talk about is work by Sean Coutts, who was a postdoc with myself and is now currently at the University of Sheffield. And what he did was we used what we know about closely related species in similar locations and what we knew about climatic conditions where populations were to build a model of population performance using information from phylogeny, from the geographic distance between populations, and from bioclimatic predictors. And the second approach I'm going to talk about is using information on climatic conditions relevant to the species niches to make the same kinds of predictions. So this is work by my current postdoc, Will Marie Curie Fellow, Anna Chergo, and what she has done is built models of species occurrences as a function of bioclimatic predictors, and then we use these probabilities of occurrence, so using species-specific species distribution models to see if we can predict population performance based on the suitability of the climate for occurrence of those species. So for both of these pieces of work, we've used the Compadre plant matrix database, which has been curated by Rob Salguero Gomez, where we had available to us demographic information for up to 1,000 species of plants. We've only used particular subsets of that database, which I'll talk to you about shortly. So the matrix models in this database have got information from populations where data was collected in the field over several years on growth, survival, and reproduction. From the matrices that are constructed, we can calculate measures of population performance, such as population growth rate, transient dynamics, and we can also look at the sensitivity and elasticity of population performance to different demographic rates. So there's a lot of very detailed field-collected data in that database. Now for Sean's work, these are the locations of the populations of over 200 species that we used from the Compadre database. And these are the colors represent the climate, a measure of climate using mean annual temperature and temperature seasonality, just to give you an idea of the range of climates that we can study populations in, from very warm, unseasonal climates all the way through to quite cooler and much more seasonal climates. And this work is published this year in Ecology Letters. So we have data on 550 populations from over 200 species with 3 years or more of field-collected data. And in only around half of these species do we have more than one population per species. And even for the species where we do have more than one population, most of those populations are within just a few kilometers of each other. So this graph here shows the maximum distance between populations within a species. You can see that there's only a very, very few species for which we have large distances between populations in order to make inferences across very large range areas. And for most species where we have multiple populations per species, the data are collected in populations that are extremely close to each other. And this is a problem if we're trying to make predictions about what populations of a species are going to do outside of the location where we have actually studied them. So what Sean did was built a Bayesian model predicting some measures of population performance, and here I'm just going to use population growth rate as a function of climate variables, so those bioclimatic variables like seasonality and temperature and a bunch of others, the phylogenetic distance between species, so can we use information from closely related species to inform on how our species of interest is doing, and he used information on geographic distance between populations, both within and between species, to see whether the geographic location gave us more information than the bioclimatic variables alone. And we did this for a number of different measures of population performance, including population growth rate, temporal variation in population growth rate, the elasticities of population growth rate to demographic processes, and the damping ratio, one of the metrics of transient dynamics. So the model looked like this. We're trying to make predictions of performance, mu, as a function of environmental variables, species variables, and then these geographic distance and phylogenetic distance functions. So for geographic distance, we were trying to fit a parameter on this exponential function to describe how quickly information from geographic distance decays when we're trying to make predictions. And the same for phylogenetic distance. Basically, we can fit a parameter on that exponential to look at how far away in phylogenetic distance can you make inferences. Now, this graph here are the results of lots of different models. And I'm just going to talk briefly about one particular result here. But on the y-axis, we have the population performance metric we're trying to predict. And on the x-axis, we have a measure of model performance, R squared here. And these are the results for different kinds of models. So this is one of the main model, including all variables for population growth rate, including some interactions as well. And you can see that we get an R squared of round about 25%, which is not too bad. If I show all of the models up here, you don't have to understand all of the detail, but just note that for some models, we get very high R squareds, and for other models, we get very, very low R squareds. So our models of population growth rate are relatively poorly performing. Models of some of the other metrics, like elasticities, perform better. And then the other thing I want you to notice here is the full model is given by this orange color, and this is a model with no climatic variables. And you can see that the climatic variables don't really add anything to the model. So when we have geographic distance, that seems to basically soak up all of the variation that could be attributed to climate. And the other reason for this is probably because different species have different climatic requirements, and a one-size-fits-all approach just doesn't work. So that's what I want you to note there. The other thing I should say about these models is that when we remove the populations which are collected in exactly the same location, our model performance goes down. So these are these no-self populations here. So in summary, our predictive capacity of geographic distance decays very rapidly, usually within about 20 kilometers. So we can get some information about population performance from other species, but only if they're very close to the location in which we want to make predictions for. And our predictive capacity of phylogenetic distance is actually a little bit better, but that again decays quite rapidly within about 50 million years. So for some metrics, such as elasticities, we can get some useful phylogenetic information. For other metrics like damping ratio, that decays very rapidly indeed. So we can make some limited predictions from just knowing phylogenetic distance and geographic distance. The second piece of work here, this approach is taking a more nuanced look at how climatic variables might affect population performance. And this is work that's currently in review. So what Anna did here was generate ensemble species distribution models from occurrence data using bioclimatic explanatory variables, using the standard bioclim variables for 14 trees and 23 herbaceous perennial species, so about 37 species in total, about 100 populations. Now, we might expect population performance, such as population growth rate, to increase with climatic suitability, but there are a number of reasons why this might not actually occur in reality. And we have examples of counterintuitive predictions, I guess, of populations. So the size of the circles here is the measure of population performance, and the colors represent climate suitability. So we have poorly performing populations here in unsuitable habitats or less suitable habitats, and well-performing in very suitable habitats. But we can find all kinds of different patterns, including well-performing and poorly performing populations in relatively unsuitable areas, and all combinations, basically. So the take-home result here is that climate suitability is, in fact, a very poor predictor of population growth rate, the variation in population growth rate, and the time to extinction. So it doesn't look like we can use probabilities of occurrence or habitat suitability to predict these sort of integrative metrics of population performance. However, we did get some really interesting results with regards to the pathways, the demographic pathways involved. So I'm just going to take you through this graph slowly. So climate suitability here is on the x-axis, and we have retrogression, which is a measure of, you can think of it as shrinkage. And we see that in less suitable climates, things are more likely to retrogress. In less suitable climates, things are likely to have quite variable fecundity. Here for trees, in less suitable climates, we see variation in growth or progression being high. So you have more variable growth in less suitable climates if you're a tree. There was no pattern if you were a herb. And then if we start to connect then how these measures of mean demographic performance affected by climate suitability and variability affected by climate suitability translate through to time to extinction, we see that the more variable your growth, the shorter the time to extinction, so you're more vulnerable to extinction. And we see here that if you can shrink, if you can retrogress, then your time to extinction actually gets longer. So that's a pattern of resistance to unsuitable climates. So we see two major pathways here. In low suitability climates, retrogression is higher, and higher retrogression is associated with longer times to extinction. So this may be a way that populations can actually resist extinction in poor, less suitable habitats. But we also see signs of vulnerability. So in low suitability climates, there's more variation in fecundity and progression, and higher variation in progression is associated with shorter times to extinction. So we see a vulnerability pathway as well as a resistance pathway in these low suitability climates. Now, this could be quite useful in terms of trying to predict what's going to happen as people make projections for SDMs going forward into the future, into future climates. We may want to investigate the vulnerability and the resistance pathways for how populations may be able to persist or may be vulnerable to extinction in these low suitability climates. So, final summary. If we want to infer demographic performance, we need to approach with caution. We can only do this currently for small spatial and phylogenetic scales if we're just using that information. Macroclimatic suitability does influence demographic pathways rather than the integrative measures of population performance directly. And positive population growth rates and extinction resistance can be maintained even in sites with low climatic suitability, and that is very much worth keeping in mind when we're making projections. And finally, we need more data. Join Plant PopNet, where we're trying to extend this axis of the distance measured between populations out to about 13,000 kilometers. It's going to be great. And we have a poster.
Timekeeping. Questions? At the back there, gentleman in red.
Hi, Hal.
I have a question. Hello.
Hello.
A question about or a thought about the small spatial scales that you found when you were looking at demographic variables like population growth rate or things calculated from it. In my experience, when plant ecologists sample multiple populations of their species, they often purposely choose different kinds of, they want the sunny side and the shaded side, the low moisture, the high moisture, to get a range of variables. And that automatically means that there's going to be lower correlation because they're actually interested in the demographic variability between those. Is there any way for you to separate that dimension of the variability out in trying to account for the ability to predict things over distances?
Sure. So we did not include any experimental manipulations for exactly that reason, I guess. But we did include all natural populations. So there are definitely some habitat differences in the data set that we used. I don't think that would affect the results. If anything, that would make the results go the other way. The fact that we found predictive performance within 20 Ks indicates that even if you do have different habitats, you're still performing more or less similarly. Because when people do those studies, they tend to be within 5 Ks, is my feeling from the data. We could certainly extend the model to look at, I think you could add in variables to account for that, basically. Yeah.
Thank you very much. I'm afraid we're going to have to move on. So our next speaker is who's going to talk about predicting species range dynamics, a benchmark test of current data-driven approaches. Damaris is from Zurich.
Yeah, thanks a lot for the introduction. I'm going to talk about going beyond species distribution models and predicting population dynamics, so it actually fits nicely to the previous talk. And the talk is mainly based on a paper that came out in the beginning of this year on benchmarking novel approaches for modeling species range dynamics, and a lot of people have been involved in that. So yeah, we want to predict not only the current distribution, but future distribution. We often assume that the current distribution or the environmental envelope will shift as the climate is shifting. And what species distribution models are basically doing, really just shifting that entire envelope in space. But that may not actually correspond to the realized future distribution, because that might be governed by the dispersal ability of the species, by persistent ability. So we might have areas where the species will persist, and we might have areas where the species may not be able to disperse to. Then we could have competitors or disturbances, stochastic events that are driving the system. And these are all things that the correlative SDMs do not consider. And the other thing is that the current distribution might also be governed by a lot of dynamics, for example, demographic dynamics like source sinks, or community assembly processes, like do we really have environmental filtering, or do we have more dynamics which look like neutral dynamics or whatsoever, that will all govern the accuracy of the models. So we wanted to compare like new modeling approaches for modeling range dynamics and population dynamics, compare these to species distribution models, and we wanted to do so under controlled conditions, which is why we chose a virtual ecologist approach, which gives us the possibility of considering various complicating factors. For example, different biotic interactions, demographic processes like source sink dynamics, high stochasticity in the system, etc. And it's actually really easy, like you simulate a virtual community, you can put into that model everything that you want, but it should be fairly complicated, I must say, at least if you want to have a fair comparison of all the other models that follow. And then you sample that, just as you would do in real world. We sampled occurrence data, abundance time series, demographic rates. And then you use this data for model building. In our case, it was species distribution models and a number of range dynamic models, which I will introduce shortly. And then you can make predictions. You can make predictions to current environment and also to the future. And now the nice thing is, because you know all about your simulated community, you can make like strong inference about how well did your model predict. And also you can run the virtual community into the future, and so you can also make like strong inferences how well the models are actually predicting the range dynamics. So this is how the model looked like. We took a multi-species grid-based individual-based model. We had different species that were described by a two-dimensional niche, and we put them in a gradient landscape where we have gradient of temperature. So initially we just distributed individuals randomly. Then in each time step, well, they could die because of normal mortality events or disturbances. And then the question is like, who will be, or will there be another individual replacing that one? And that will be a function of offspring production and seed dispersal, but also seedling survival. And both these processes are described by the niche of the species. And yeah, this is how it looks like along one niche axis. Of course, we had a two-dimensional niche. And in most cases, the niche for survival and reproduction will be the same. But we can also have cases where we have like a broader survival niche, which would allow us to simulate source sink dynamics. But then the species is not alone in the landscape, but we have other species competing with that species, and the other species could either have exactly the same niche, which would then result in neutral dynamics, or we could have some sort of environmental filtering, some species sorting. And then it's a question of the reproductive value that the species has in that cell, and of how many seedlings come, and that will, well, randomly determine which species will win the race and which individual will be placed there. So by systematically varying those community and demographic processes, we have different scenarios. We have species sorting versus neutral dynamics versus just a single species scenario. We consider source sink dynamics, different dispersal abilities, and some stochasticity in the system, which we simulate by contagious disturbances. And then we let the model run. We can apply climate change, and in the end, we can sample the focal distribution. The problem here is that we have an individual-based model with individual locations, and in the end, of course, the range dynamic models are working with populations, so we're introducing actually a second scale where we just aggregate like 10 by 10 cells, so we have per cell we have a maximum of 100 individuals that could be there. This is how the simulated range dynamics look like. So we see some nice time delays. On the left-hand side, you see an example for the long-range dispersal. On the right-hand side, for the short-range dispersal. And in blue, you see like how the mean abundance is distributed along the temperature gradient for the current year, for the observation period, and in grayish, orangish, you see how it looks like after climate change. So we do see that we have some dispersal limitation at the cold range front, also for long-range dispersal, but more severe for short-range dispersal. And we also see some persistent at the trailing range edge, at the warm edge. And we also see some distinct spatial patterns. So this is an example for neutral communities, again, for long-range dispersal and short-range dispersal. For the long-range dispersal, we have the species, well, this is really just the focal species, the abundances, like with darker colors indicating higher abundance. And we see that for the long-range dispersal, the species is more or less evenly distributed in the range, while we have a much patchier distribution for the short-range dispersal, because of stochastic competition. And we also see that we have more variation in local abundances, for example. And this leads to quite different patterns under climate change, where the long-range dispersers are more or less shifting their range. The short-range dispersers experience severe range contractions because it's very stochastic, whether they can move or not, depending on whether there is a population in that patch or not. So we tested 5 different models, the correlative species distribution models against 4 range dynamic models, 3 of which were SDM hybrids. It's not a very nice name, I know, but it's basically saying that they still use some output of the SDMs to inform the population models. And then last, we have the dynamic range models that completely avoid SDMs. So for the SDMs, we used an ensemble approach. using different methods, maximum likelihood, machine learning, etc. And they output like habitat suitability maps and or presence-absence maps. And these are used as input for the hybrids. The most simple hybrid is MICLIM, which just supplements the habitat suitability maps with some measure of dispersal. With the dispersal kernel, there is no automated fitting, so you really have to assume how the dispersal kernel would look like, and in the end you get a prediction how likely it is that a cell gets colonized or not. And Demonit is a matrix population model. A more popular one is probably Rammer's GIS that you might know, and that basically couples the SDM output with the dispersal kernel and the matrix population model, and there you have to make decisions like whether you scale the demographic rates by habitat suitability or, for example, carrying capacity. And in the end, you get the population dynamics. So in blue, you see the predictions of abundance, and these are, of course, driven by the processes dispersal, birth, death. So you have much more process in there than in the MICLIM model. And in that case, we optimize the demographic rates for calibrating the model. And then we have Lollipop. That's a maximum likelihood-based approach. It takes the Beverton-Holt model, so some form of logistic growth model, including alley effects, as population model. It only uses the presence-absence maps from the SDMs and the dispersal kernel, and tries to optimize the spatial abundances to calibrate the growth rate, basically, in the population model. And it can also simulate population dynamics in the end. And the last model are dynamic range models. These are fit using a hierarchical Bayesian approach, and they completely avoid this SDM output, but they directly relate the intrinsic growth rate of the species against environment. So they have a species demography relationship in there. They take in that instance, they take the simple Ricker model, so logistic growth model. And they also estimate, they also calibrate dispersal all in that Bayesian framework. Results. So luckily, we found that all range dynamic approaches improve predictions compared to SDMs. So the row is the correlation between the predicted and the observed abundance on the left-hand side for the current climate and on the right-hand side for the future climates. And the SDMs are making far worse predictions than all the range dynamic approaches, which is good. But we also see that on average, the prediction accuracy decreases with climate change for all models. And this is mainly a problem with predicting at the leading edge, for example, because of biotic interaction. So in the species sorting scenarios, the competitor species are acting as some form of dispersal barrier, which is not predicted well. And in neutral communities, we have this high stochasticity through the local clustering. And in these cases, actually, the simple dispersal models often work much better than the population dynamic models. Then, of course, some of the models can predict population dynamics, and we see some interesting pattern there. So up there, you see the abundance trajectory for the period of climate change, and the solid blue line indicates the observed abundance. And we do say that Demonit with a red dashed line is actually overestimating abundance quite a lot, while the trajectory looks better for Lollipop and DRMs. But when we actually look at the relative change in abundance, so really how the population trajectory looks like, then the picture is quite different. So here actually the matrix population models are doing better. And maybe don't look at the box plots, I don't have time anymore. But it's mainly a difference whether you calibrate on the abundance data or on the demographic data. We also looked at structural uncertainty. As I said, in those hybrids, you have to make decisions like how do you scale the habitat suitability with the demographic rates. And I mean, here you should ideally have accuracy of 1.0, and we see that accuracy is decreasing quite a lot and differently for different configurations. That's an example for MICLIM. And we see the same for the matrix population models, but I'm not going into that but you can look at the paper. So the main conclusions are that dynamic approaches do improve the range predictions over simple SDMs, which is good. However, we could not identify any like one best single approach. What we already know from population viability analysis, it has been said in the 90s, that often predictions of absolute abundance are not very accurate, while predictions of relative abundance are much more reliable. We also found that. Then third one is that, of course, if we do have differences in dispersal ability and complex biotic interactions, that can introduce high uncertainty in the range predictions. But we've also seen that if we do have some prior knowledge on the system, which is what I haven't shown you, but we can reduce the structural uncertainty if we know something about the system prior to modeling, which is also good. But I think in the future we should really work more on merging current modeling approaches. So clearly those dynamic range models that use the joint fitting of the species demography relationship, they're much more grounded in ecological theory than those hybrids that use the SDMs, and we don't really know how and why and whatever. So we should really work on merging those approaches, because at the same time the matrix population models, they offer much more flexibility and modularity, which is better. And we should incorporate interspecific interactions and, of course, compare against field data and look at other complicating factors like sampling effort and so on. Well, thank you.
Questions?
Sorry. Given that all of the dynamic ones basically perform the same, are any of them markedly easier to fit or use or can parameterize than the other ones?
Yes, definitely. Well, I mean, they're not predicting exactly the same. They do have some differences. It's just on average, we could not identify one single best. But of course, MICLIM, I mean, you don't need any fitting. It's the easiest, but you do need to have some idea about how the dispersal would look like. And I think the matrix population model and the lollipop are still easier to fit, of course, than the dynamic range model, because it uses quite a lot of computing power, and not everybody is able, I think, to parameterize a population dynamic model using Bayes, so that's quite a restriction at the moment.
Okay, thank you very much indeed. I'm afraid we need to keep moving on. Our next speaker is Charlie Boothway from UCL, also working at CEH and with the RSPB, and her talk is about the other taxa behind the State of Nature 2016 report, exploring the results from biological records.
Thank you very much. Hi everyone, yes, I'm Charlie. I'm a PhD student at the Centre for Ecology and Hydrology and also an author on this year's State of Nature report. So I'm going to talk to you today about my contribution to the report, which involved estimating species trends from biological records. So the first State of Nature report was published in 2013 as a collaboration of around 20 organisations. Its main aim was to provide an overarching assessment of the status of UK wildlife. And it aims to complement other measures, including the UK biodiversity indicators, in quantifying this kind of change over time. As you may know, the UK biodiversity indicators, specifically for species trends, consist of the wild bird index and butterfly indicators, so two major groups for which abundance data are available, but this doesn't cover a wide range of species. So the State of Nature report really broadens what we're able to know about UK biodiversity. The report was published again this year, this time as a collaboration of over 50 conservation and research organisations, and through advances in the way we're able to analyse the data that we have, we've been able to include more taxonomic groups and more species to this report. And the report has been really influential in shaping the debate around conservation and biodiversity research within the UK. So there are two main statistics within the report this year. The first, shown here, consists of species trends for around 3,800 species, and it shows that over the long term, so between 1970 and 2013, 56% of species have declined, 44% have increased, and over the short term, the most recent decade for which data were available, from 2002 to 2013, 53% of species have declined and 47% have increased. The second statistic was an indicator in the change of species status, and this was composed from a subset of those species included in the first statistic, so 2,501 species. Not all species had annual estimates available, so this is a smaller amount of species. And what this showed was that on average, a species had declined by 16% since 1970. The species included here had their underlying data derived from various sources. So 40% were from abundance data. So again, that would be the kind of birds and butterflies that we're seeing in our main UK statistics. And the other 60% covers a much broader range of taxonomic groups. So things like beetles, flies, bryophytes, and things like that. So a much broader taxonomic coverage than we're able to get from abundance data alone. And those are the sets of species I'm going to talk to you about today. So our data for the occurrence records are biological records. So these are presence-only data, usually collected by volunteers as a part of a recording scheme or society. But due to the unstandardized nature of the data collection process, there are a number of biases associated with this. So if I give an example, perhaps a volunteer interested in hoverflies, he'll perhaps just go out at the weekend to a spot where he knows he'll find some hoverflies, and he might note down everything he sees, he might note down just those few species he's interested in, and then he'll submit those records to the hoverfly recording scheme. However, a lot of data is available in this form, and through my position at BRC at CEH, I've been able to gather data on 7,500 species from 20 taxonomic groups, shown here. So you can see there are no birds or butterflies here. We've got groups such as ants, bryophytes, millipedes, and grasshoppers. So to analyse this data, we use Bayesian occupancy models. These are hierarchical models that separate out the ecological process underlying the true occupancy state of our sites from the detection process underlying the data. And this is really important because we need to account for imperfect detection. So just because a species wasn't seen doesn't mean it wasn't there. By organising our data into detection histories, as kind of exampled at the bottom here, where we use our records of a focal species to infer detections, and records of other species and non-detections of our focal species to show non-detections, zeros here, we're able to use what would otherwise be presence-only data to analyse, to use occupancy models to analyse what would otherwise be presence-only data. So I fitted models for each of those 7,500 species. They were run by taxonomic group, and each group took between a few hours to a few weeks to run on a computer cluster. So it's quite computationally intensive. When we started looking at the outputs from these results, we saw there was a lot of variation in kind of precision around our estimates. So I've got two ant examples here, and you can see that the one on the left, we can be quite confident around the majority of our estimates, bar the beginning and end of the time series. And on the other side, the probability of occurrence can pretty much be anywhere between 0 and 1, so it's not really very helpful. So we did have to make sure we separated out those we considered reliable from those more messy outputs. And after doing this, we did end up losing 80% of our species outputs. So my results coming on from this will be shown using the subset of 1,604 species for which reliable outputs were produced. So this is an indicator of those 1,604 species, and looks at the change over time since 1970. And what you can see here is that there's been perhaps a slight decline towards the mid-80s, and then the majority of the change has occurred in 1987. Since then, levels have been quite stable, but overall, on average, a species has declined by 15% since 1970. If we look at species trends here, we've got the balance of the proportion of species increasing and declining shown by taxonomic group, where the number in brackets is the number of species included. So here we've got the proportion of increasing species in blue and the proportion of decreasing species in red and orange. So you can see there's a lot of variation here between and within taxonomic groups, and you can pick out certain groups for which more than 50% of the species are declining. So we've got crane flies near the top, empid and dolichopodid flies, orthoptera and soldier flies further down. If we look at these taxonomic groups more closely, we can see where this change occurred. So the majority of taxonomic groups showed similar responses to those shown here. So quite strong declines in the early part of the time series, so between the 70s and mid to late 80s. and then relatively stable responses since then. So here we have the empid and dolichopodid flies. N is the number of species included within the indicators here. And they've shown a decline of 45%. The Orthoptera and allies have also shown a decline of 53%, although there's a lot more uncertainty around the estimates here. A few of the groups looked relatively stable or tended to hover around the baseline. So we have the bryophytes and lichens showing examples here. These were the largest data sets that I analysed, and this kind of reflected in the number of species that you have contributing to the indicators. And the kind of more tight credible intervals are a function of that increase in the number of species included. An interesting response was shown by the caddisflies. So you can see here that they've undergone a strong decline between 1970 and the mid-90s, and then since then have had a quite strong recovery. This was the only group that showed this kind of response, so I did get a little bit excited by this. So they reached their lowest point in 1994, and since have shown quite a strong recovery. Caddisflies are an indicator of water quality, and a lot of work has been done within the UK to try and clean up freshwater systems. So it's quite possible that this is being reflected at a national scale. Dragonflies are also a predominantly freshwater group, and you can see there's a kind of similar response with declines up to the mid-80s and perhaps a very slight recovery since then, but this isn't as prominent as that seen in the caddisflies. So here I have split my 1,604 species into two groups. So I've got priority and non-priority species. So within the UK, we have a priority species indicator. It's one of those published annually. And it focuses on species that appear on one of the 4 country biodiversity lists. So this contains species that are usually small, ranged, rare, or declining. So as you can see from the indicator, probably get what we would expect in that priority species have undergone a greater decline than non-priority species, but the kind of general trajectory is very similar. So overall, hopefully I've been able to show you that biological records have really broadened what we've been able to learn about UK biodiversity. There's a lot of variation responses between species and between taxonomic groups, but we've been able to show that overall wider UK biodiversity has declined by 15%, and so particularly looking at these species that little is known about, rather than focusing on the birds and butterflies that we know quite a lot about. As you would have seen, I did have to drop 80% of my outputs from the final results, which is rather disappointing. So since then, I have been working on improving the modelling techniques that we use to make better use of the data. So these are groups which quite low levels of data are available. So making the most of what we have is really important for carrying this work forward. And so far, this is looking really promising. I've been able to get much more precise estimates out for a large number of species. So perhaps next time I'll be able to show you trend estimates for 7,000 or 8,000 species instead of just 1,600 or so. Just a quick thank you to my supervisors and the other people I work with at CEH. Of course, the State of Nature team, which helped me contribute to the report, and definitely to the volunteers who go out and collect these masses of data in their free time. So thank you very much.
Charlie, so we've got quite a lot of time for questions, beginning here.
Hiya. Hiya, that was really interesting, thank you. So you talked about some of the biases with species being less sampled or people focusing on some species. Are there any habitat-specific biases? So maybe people are less likely to sample in degraded habitats, and could that affect anything, and is there a way to account for that?
Yeah, I think that's definitely an issue. I mean, just speaking more generally, where people record and kind of spatial biases over the country is quite a problem, because we find people generally record species near where they live, and people don't go too far out of their way. I mean, there are some very keen people that will try and fill in the blank squares, but yeah, spatial biases is something that's quite an issue. Within the models we're looking at the moment, we do include a site effect to try and account to some extent for that, but I think, yeah, it does need to be looked into more. And I think it's also an issue when you're looking across so many groups that it can vary quite a lot by group and by scheme exactly what those biases are. So it could be tough.
Thanks. It's a really interesting technique. I suppose I want to question how far can you push this? Is it really worth trying to go for those 7,000 or 8,000 species? What I'm thinking is, for some of these, the number of experts who can record these reliably is tiny, and you may actually be, recording trends in expertise of amateur natural history, and a decline in biodiversity is actually a decline in taxonomic expertise amongst the amateur naturalists. I know you try and take account of recorder effort and so on, but I suppose the bulk, to put it more punctually, should you be going and looking for more and more rarer and rarer species, or putting more effort into accurately monitoring? Okay, sorry, Steve.
I think it's important that we monitor all these kinds of species, and the ones I'm looking at aren't particularly all rare or small range that we should be spending lots of time going and looking specifically at these. A lot of them are really common species that we just don't know a lot about because there's no kind of standardized data collection process for those species. I think there potentially are issues around kind of recording, but the number of records over time for all of these groups has increased dramatically. So I think, I'm not sure, perhaps if there is a chance of measuring something that's a little bit off, it would be more likely to be an increasing trend because we're getting so many more records than previously. But these schemes do work across a range of people, and the range of expertise does vary, and there's a lot of kind of support out there for them. So I think they're maintaining what they do quite well, and it's just up to us to make the best use of what we've got.
Thank you. One sentence, please.
Okay. Charlie, thanks for that. 53% up, 47% down sounds pretty good if you're a policymaker. That's 50-50 is what you'd expect if it was average, wouldn't it? Mm-hmm. What do we know about abundance of nature changing? Biomass, perhaps, or other measures that actually might better describe what's really happening?
Yeah, I think the whole point of using this source of data and trying to make the most of this vast resource is that we don't know a lot about the abundance of the majority of the groups I'm looking at anyway. Those species that we do know, or we do have abundance data for, obviously again those birds and butterflies, and lots of work has been done on indicators and trying to figure out why these changes are occurring. So where abundance data is available, I think it's really important we take advantage of that. But where it's not available, I think you've got to make the most of these alternative sources and try and figure out what's going on from what you've got.
Thank you very much, Charlie.
Thank you.
From UCL, and he's going to be talking on including biotic interactions and models of species distributions and assessment of extinction risk.
We don't have a laser. Sorry.
Okay, thank you very much. So actually what I will do is somewhat pick up on a couple of talks ago, the Zurl talk, which was fascinating. I'm going to build on that. So one of the things that was left at the end of that was this idea of trying to build in species interactions, interspecies interactions, biotic interactions into some of these macroecological models of species distributions. I should emphasize, so I'm Richard, the last on that list. Philip Staniksenko is really the lead on this work. He's been a postdoc in our center for a couple of years out in the US at the moment. He's done a lot of the Bayesian analyses that I'll talk about. And then Blake Suttle in particular as well is has the knowledge of the ecological system, the Californian grassland, that I'm going to use as a brief case study. So very briefly, why are we interested in modeling species distributions? Well, of course, there are a whole bunch of questions that we can address in terms of trying to predict new populations, reserve planning, predicting invasions, spread of zoonotic diseases. The list goes on. I won't go through it anymore, other than to say one of those is predicting impacts of climate change on biodiversity, on species distributions, on abundances, on extinction risk. And there's a little bit of bias in my next few minutes in terms of using that as the case study, but really the kind of framework that we're trying to work with and present is very much about just in general trying to predict species distribution. So how can we do that better for across a whole suite of different types of applications? So let's step back to some of the basic theory here in terms of asking, well, if we're interested in modelling distributions, what are some of the factors that really define where we would find a species in the landscape? We often work with what we term the BAM diagram, so the biotic elements, the abiotic elements, and movement. So these different factors that determine where you would expect to find a species. So, of course, the biotic environment, the community that species occurs in, symbiotic relationships, competitors, the presence of prey, these are going to define where you find a species. The abiotic environment, temperature, precipitation, soil type, any number of variables that define the abiotic environment where we would expect to find a species. And then movement. So where the habitat and the community are suitable, then what's the potential for that species to actually move around the landscape and occupy those particular areas. So the large field of species distribution modelling that we've heard about even just in this session already essentially focuses on the abiotic environment. So these are methods where we take what we know about the species distribution, we take some information on the abiotic environment, so temperatures, the bioclim variables that Yvonne Buckley spoke about in the first talk, things like minimum temperatures, maximum temperatures, precipitation regimes, but then all sorts of abiotic variables, soil type, distance to rivers, these kinds of factors. So we build up a statistical relationship between the two and use that to define where we would expect to find the species. And that's been applied really widely, think methods like Maxent, Biomod, and the like. But they focus on the abiotic environment. Now, a big step, I think, in the last 5, 6, 7 years or so, again, that we heard a terrific review a couple of talks ago of this whole field, I've termed here coupled SDM or species distribution models with demographic models. I think in the talk you referred to it as linked models, but essentially the same principle, where we take, again, the abiotic environment and what that defines about the species distribution, and we link that with a model of movement. So dispersal ability, probabilities of dispersing to a given distance, and the population dynamics that enable species to move through the landscapes. And as has been shown in a number of studies, these are methods that can help to build better predictions of species distributions. But what we're still missing in most of these kind of macro scale predictions of species distributions is the biotic environment. And we're still doing a very poor job, I think, in this field as a whole. And again, it was flagged a couple of talks ago as one of those things to move on to in terms of how we can start building interactions with other species in the community into these predictions of species distributions. So what I'm going to just spend a few minutes talking about is an approach that we've been testing and working with an interaction network model to do exactly that. It's relatively early days. The manuscript that we're working on is about to be resubmitted. So it's early days for this work, but I'm trying to set up a framework that we can start using. An important part of this picture is spatial scale. So as a macroecologist, and most of these field in terms of species distribution modelling and coupled SDMs with demographic models, are thinking large scales. These are macro scale ecological models, where we tend to think, well, the abiotic environment and movement are the most important factors. But those of you who have maybe come out of an interaction session or a community ecology session at this meeting, stumbled into a session like this, we'll be hearing less about community ecology, and we're tending to think about different spatial scales. So what's happening at a much finer spatial scale? Well, here's just one example from my co-author Blake Suttle's work. He's worked for more than a decade now in a Californian grassland system where he's experimentally manipulating the climate, in particular the precipitation. So he has very good knowledge of the ecological community in this system, and what he's shown, including in this science paper back in 2007, is that the responses of species to environmental change are driven by species interactions. So it's those biotic interactions, it's the interactions with other species that are driving the response to environmental change. Yet, of course, as a macroecologist, when we, this is a bit more of a cartoon example, but when we think of some of the work, in this case in North America, Camille Parmesan and others who've studied distribution shifts, at a macro scale, we get these much more consistent and somewhat predictable patterns, for example, of northward rain shifts. So what's going on here? How can we kind of marry this, what's going on with the community interactions at a very local scale, with what's happening at a macroecological scale, where we have these complex local dynamics driven by biotic interactions versus these relatively predictable macro scale shifts in distributions? So really what we're trying to do is set up a framework where we can somewhat get these two communities and data sets and methods talking to try and link across these different spatial scales. So working with this grassland community, particularly Blake Suttle, co-author, we've worked to build up an interaction network. It's very simple, it's very straightforward, it's incomplete. We've focused on these 7 plant species that you see in that central ring there, and we've tried to define just positive and negative interactions to build up this network of interactions. As I say, it's incomplete, but it's some on-the-ground experimental knowledge of what the community looks like. Then what we do is use a classic distribution model. We've used Maxent, but you could use any model, any distribution model, to generate what are essentially priors in a Bayesian framework, but they're just predictions of probability of occurrence for each individual species, so relating the species to the abiotic environment. Classic thing that's been done very, very widely in the literature. Then essentially we throw that, we use Bayesian networks, classic kind of method for looking at how those probabilities will interact within the network and bounce around, so you have positive and negative interactions within the network. So we feed the individual species models into the network and then get out our posterior probabilities, which are not just what's the probability of a species occurring based on the SDM, the abiotic environment, but this also takes into account what's happening to changes across the network to all the other species. In terms of results, as I say, it's early days, but what we can at least show is that there's some promise here, that we're being able to predict at least the present day distribution. So this is just a Monte Carlo cross-validation of the present day data that we have across those 14 focal species. And on your x-axis here, we're seeing just the difference between the posteriors and the priors. So when we take the interaction network into account versus when we don't, and the shifts aren't huge, but we're at least showing that everything above the 0 line, the red line, is an improvement in model performance. So we're at least showing that we can get some improvement in model performance, and it's consistent. We can then do things like predict, in this example, under a future climate scenario. So we do a classic kind of prediction. The green here, again, this is for kind of the area around California. The green is the prediction of where the habitat is suitable. We can predict forward to 2050. If we interpret these shrinks in suitable habitat in terms of extinction risk, which is something that's, for better or worse, widely done in the literature, we can show that when we include the interaction network, the loss of habitat, or the extinction risk, if you like, is around 65%. Without the interaction network, it's about 59%. So not huge changes, but we're being able to show that by taking the networks into account, we get somewhat different predictions of what the extinction risk is likely to be. Now, as I say, it's early days. This is just across 14 species that we've worked with so far. Across those 14 species, we're most often finding that the biotic interactions lead to reduced estimates of extinction risk under environmental change, which is, of course, one of the kind of headline things we'd like to come out with in time. You know, does including the networks, including the interactions, increase or decrease estimates of extinction risk? I'll emphasize that it's, you know, I'm not making this claim at the moment. It's too early to tell. But what we're doing is setting up a framework where we can at least start dabbling with some of these things. So there's a whole bunch of new things that we need to do. A classic thing is we need to test across a larger sample of species and different communities. So we're looking for test systems where we have known interaction networks, the kind of system that I've shown. So I'd love to chat with people who have systems like that. But at least I think we're able to show at the moment that we have these kinds of methods hold some promise, and co-authors and funders that I must thank. Thank you.
Thank you, Richard. Questions? Again, Mike. Short, please, Mike.
Species distributions are, or realized distributions, take account of climatic interactions. Species are there partly because of climate, partly because of interactions. Is it therefore not already implicit in the original SDM?
Yes, so to a degree, and there's a lot written on this, and SDMs, as you say, are based on the realised niche, so there is some element of interactions being taken into account. One of the things we'd like to do with these methods is build the distribution models in a much more mechanistic way so that we're trying to get a better estimate of what the fundamental niche of these species are. And then I would expect to see a hypothesis, if you like, that prediction, the improvement should be greater if we were to use methods like that estimate fundamental niches. But it's a very good and valid point to raise.
Any other questions? Yes, here in the front.
Is there also the possibility of...
Can you use the microphone so everybody
Yeah.
hear?
Sorry. In your network approach, as I take it, is that you, the species interaction coefficients, you just take them from the experiments, or do you also calibrate them? Because across the range of the species, it could, of course, be that the interaction, for example, varies with the environment gradient.
Yeah, absolutely. So we don't weight them at the moment. We don't change them across space or time. But those are exactly the things that within this framework, if we've got empirical data to parameterize the models, we could do. So you're exactly the kinds of things that we would like to do. Maybe we'll talk about that in 2 years' time, or 5 years' time, or 10 years' time. But yeah, exactly.
Okay, thank you, Richard. Our next speaker is Alma Strandmark from Stockholm, and she's going to be talking on drivers behind local and regional arthropod diversity. And there you can see the full title. Thank you, Alma.
Thank you. Yes, I'm a PhD student from Sweden, and my PhD work concerns climate change and Baltic shore ecosystems. And this is a part of my thesis that I will present, hopefully, in June next year. And it concerns, well, the drivers behind diversity at different scales in a naturally fragmented landscape. And we see it as a base for future predictions on climate change and biodiversity loss in these systems. So fragmentation is a huge driving force controlling species diversity and abundance by controlling area and connectivity between habitats. And both habitat fragmentation and climate change have been said to be likely to increase global homogenization, with species, more generalist species, being more widespread over the world. So in this study, we used this naturally fragmented landscape, the Stockholm archipelago, which encompasses about or more than 30,000 islands. And within this fragmented system, we have looked at arthropod diversity in shore meadows, and we used different spatial scales. And we've done this because increased sea level rises will increase fragmentation in this already fragmented landscape. So this is the main theory behind it. We have sea level rise, increasing fragmentation, decreasing area and connectivity, and then the question is, what happens to species diversity? Oh, no, maybe that comes later. Okay, so this is the typical shore meadow system in the Stockholm archipelago. It's usually small and interspersed within this land, rocky and forest and water. So it's fragmented in different scales as well. You have the small habitat within the landscape, and then you have the islands. So lots of fragmentation going on there. So what we've done, we've used 13 island landscapes within the Stockholm archipelago that you can see here. Over there, that's, well, the red circle up there shows the vicinity of Stockholm, the capital city of Sweden. And in these 13 island groups, we have 45 islands, and on each island we have several shore meadows. So altogether, more than 140 shore meadows have been investigated. And we looked at plant diversity, and we used this converted leaf vacuum blower to collect insects and spiders. And as I said, we used different spatial scales. So if the red dots here represents the shore meadows, one shore meadow represents the alpha diversity, while shore meadows or diversity in shore meadows on the whole island represents the gamma diversity. And then we have the difference between the local species pool, which is the beta diversity. We looked at spiders and beetles, and we also divided them into different functional groups based on species traits. So we have predatory beetles, we have herbivore beetles, and we have the group other beetles, which is mainly detrivores and omnivores. And then we have, for spiders, we have the large group wolf spiders, which is the most common large predatory group in these systems. And then we have a group called other spiders, which is basically everything else. And since this is a nested design with data nested within island and island group, we used mixed effect models to look at this. And as an environmental predictors, we have both local site conditions like plant diversity and habitat size and exposure. And we have within landscape variables like the proportion of suitable habitats within the near vicinity and habitat heterogeneity. And also classical island biogeography predictors like island size and distance from colonization site. And island landscape variables, which means, well, the number of islands in the near vicinity or in the island group, and also the proportion of different sizes of islands. But right now, I will focus mainly on area and connectivity because the results are kind of complicated and I won't have time to go into every detail, but within this model, we have area and connectivity as the most important factors that are affected by fragmentation. So for the results, area and connectivity. And as connectivity here, I've used the island landscape variables, the proportion of different sized islands and the number of islands within the island group. And area is both island size and habitat size. So they are kind of grouped into two different groups of variables. And then we have the local diversity, the regional diversity, and the beta diversity. So the main patterns are that area mainly affects the local and the regional diversity, while the island landscape mainly affects beta diversity and regional diversity. So we have a positive relationship between larger area and local and regional diversity. And for island landscape, we have mainly two variables that are affecting diversity: the number of, or the proportion of medium-sized islands and the number of islands, which gives a positive effect on beta diversity and regional diversity. So this will mean if area and connectivity affects diversity in a positive way and fragmentation decreases area and connectivity, then we will have a decreased diversity generally at all spatial scales. Oops, that was a bit fast. Well, what happened now? Okay. So this pattern is kind of general for all these different groups, but mainly for these three, the predatory beetles, the other beetles, and the group other spiders. While these two, the herbivore beetles and the wolf spiders, are reacting kind of differently. So if we start with the wolf spiders, although we have area influencing both beta diversity and regional diversity, the main thing is the island landscape. And in this case, it is the number of islands, but it's not the proportion of medium-sized islands, but rather the proportion of small islands within this landscape. So we have a positive effect of a large proportion of small islands at all levels of diversity, which will mean, if we speculate on it, fragmentation would give us a lower number of islands with rising sea levels, and possibly a larger proportion of small islands as island size is decreasing. So for wolf spiders, we have kind of both a positive or negative effect when the number of islands decrease, but also it might be a positive effect if the number of small islands increase. So for the herbivore beetles, we also have some effects of, there should be a couple of plus signs there that have disappeared. But we have a positive effect of area and island landscape, but the main thing here is, kind of obviously plant diversity. A high plant diversity can support a higher diversity of herbivore beetles. But plant diversity is also affected by area, so we have kind of the same picture here with fragmentation affecting area, affecting plant diversity, and a lower local and regional diversity. So main conclusions are that local diversity is generally affected by area, while the regional diversity is influenced by area and different features of the island landscape, while the beta diversity is mainly influenced by features in the island landscape. And we will have effects at all levels of diversity by sea level rise and further and future fragmentation. And herbivore beetles will kind of be affected in the same way as plant diversity is affected, while the picture for wolf spiders is a bit more unclear. And what we will do now is that we will start to work on a species distribution model using some species from this data set and some additional species from another data set, and try to scale this up from these island groups and look at what might happen in the whole Stockholm archipelago as sea levels are rising. So we will make different projections on different magnitudes of sea level rise. Oop, wrong way. Thank you.
So questions from?
Hello, thanks. That was really interesting. I was just wondering about your sampling strategy.
Yes.
Was it area independent, or did you increase your sampling effort proportionally with...
We did not. It was the same sampling effort for each site.
All right, brilliant. Thank you.
Other questions?
Hi there. You talked about using mixed effects models, but your result, which generally you have one response variable, one predictor variable, but the way that you presented your results, you had some indirect effects and you had several different types of diversity at the bottom. So I was a bit confused about the relationship between the models. Were you talking about lots of different models there, or were you modeling indirect effects? I couldn't quite...
You mean the mixed effect models? Or, well, what we did was that we basically put everything that was not correlated into the same model, and then we removed variables with the highest p-values until we had just some of them left. But I don't know if that was your question, really. Sort of.
Maybe you could follow it up afterwards.
Yeah.
Oh, I have a suggestion. Then maybe because you have a lot of interconnected variables, or that you wanted to disentangle and their effects on biodiversity, maybe you should have considered using something like a structure equation modeling, or if you have a mixed models, then a piecewise structure equation modeling, and put them all in the model, and then somehow through model selection process get down to indirect and direct effects and so on. Maybe something that you should consider to do, I think.
Have you also, or are you planning to look at dispersal ability, for example? The different insects might have very different abilities to colonize those different islands?
We talked about it, yes. And now, I mean, I will finish this spring, so
there's no time
I
left.
probably want to do that, but if I get some additional money, perhaps.
Yeah, it's a fascinating system. It's really cool.
Yeah, it is, and it's kind of, well, diversity and abundance of arthropods in these systems are kind of unknown so far, so it's kind of new data, which is fun.
I've got a question, if I may.
Yeah.
This may be a very noddy question, but sorry, thank you. It looked as if there might be a positive correlation between size of islands, small islands, and number of islands. So I was a little bit concerned about that, just looking at your map, you know.
It is kind of a correlation, but not a significant one. But yes, I have thought about that as well, that number of islands could be influencing also the small island effect, or how to put it. But I have looked at it.
Fine. Well, thank you very much indeed.
Thank you.
She's talking on gaps or traps. There you are.
Hi, thank you very much everybody that's not just left the room. And I'm going to take you on a quick tour of the marine intertidal environment. When we're trying to look at where species distributions extend in the marine environment, we come across some major biogeographical challenges at the start. And whilst we're all concerned about the impacts of global change on biodiversity, the methods that we have to translate these broad-scale shifts into climate, into direct impacts on living animals, still remains simplistic. So when we're trying to model species responses to climate change in the coastal environment, we're really looking for multi-annual distribution and abundance data. We really need data from large sectors of the biogeographic range. And also, we need correct and current taxonomy, which isn't always the case for marine species. Now, at least OBIS is a really, really good start globally to try and address some of these problems. And again, because we're very interested in imparting all of our information to policymakers so that they can use it in conservation, we've started to notice there's a huge amount of difference in the range of data availability globally. And this can be due to many reasons: self-funding, historical activities or lack of, different socio-economic environments, and political priorities. So we don't have the information all over the planet for these species that we would like to. Here's just a quick example from OBIS. Now, the top there are all the data records in OBIS for mollusca. Now, obviously, I'm not the only person that likes mollusks. A lot of people seem to. Everybody's collected data on them. This is great. Poral bryozoans are obviously not quite so cute, charismatic. There are plenty of bryozoans around the world, but unfortunately, there's not an awful lot of data on them. So it's really, really dependent on the taxa that you're studying as well. And this is just a quick example to try and highlight that point. These are all the OBIS data points around the planet here, and you can see where the hotspots for data collection have been. And is it just a coincidence then that in these same areas, this is where the major climate-driven range shifts in the marine environment have been published as well? I'm not sure. So I think that we, as a marine science community, really need to start addressing these knowledge gaps around the planet. Now, I can't tackle the entire planet myself, so I'm trying to be content with Northern Europe. I'm the lead for the Marklin project, And this looks at the abundance and distribution of 85 intertidal species of macroalgae and invertebrates around the British coastline. Those are our sites that are marked in red there. And this is my lovely team around there as well. And Marklim is actually the most spatio-temporally extensive intertidal time series in the world now. We're fortunate we had really insightful ecologists collecting data back to the 1950s, which is great. And I've been collecting data annually for all of these sites since 2001. And we've detected some of the fastest biogeographic shifts in response to climate change in any natural system on the planet. So we actually are doing a reasonably good job, but again, in a small geographic area compared to the global problem. Now, before I move on, I also just want to raise the issue of climate change, because we keep hearing, oh, the climate's warming, this is happening here, you know, everything's getting warmer. But the map on the left here from NOAA shows that for the marine environment, it's not warming at the same rate everywhere, and there are actually parts of the global oceans that are cooling, including that patch just up to the left there of Britain. And some of the reasons for that are that the Gulf Stream system is weakening, and the Atlantic meridional overturning circulation is slowing down. This isn't new. Marine scientists have been speculating this since the 80s. However, suddenly we've got the data available to show that it's happening. And I think most concerning for me as a marine conservational scientist is if we get moderate to strong global warming, there's an up to 65% chance that this global circulation in the oceans will completely shut down. And who knows what will happen to biodiversity then. But I'm sure you've all had a really nice time here, so I'm just going to bring the mood down a little further. Climate change isn't the only problem for our species. We're also having to deal with ocean acidification. And I'd just like to show this example here because it's a friend of mine, Brian Helmuth, who's done this in America. And he's shown that not all species are actually having negative interactions with climate change and OA. This lovely sea star here, Pyzaster, actually has a positive response to ocean acidification, and because what it does is it fills itself up like a reverse hot water bottle, so it fills itself up with cool seawater before it goes predating in intertidal. It can actually deal with climate change and elevated temperatures as well. So we all thought this was a super species, you know, it was going to overtake the coastal zones around the world. Oh, but then wasting disease came along, so that's completely wiping out the species at the moment in the Pacific. So there's another problem. And just to depress you even further, especially for the coastal zone, but in all global oceans now, microplastics are starting to become a real issue. And we're seeing concentrations increasing in coastal waters. They accumulate in a lot of filter-feeding species that are either commercial and or keystone species in benthic environments. And I think more concerning as well for people like me is that there is a synergy between stresses as well. So we're getting completely unprecedented rates of change with climate change, ocean acidification, and then microplastics as well. So if you are especially a filter-feeding invertebrate living in the coastal systems, it's really not looking good for you at all, unfortunately. So how do we try and work out then, biogeographically, what all of these different multiple stresses are going to do to our species in terms of distribution ranges? And we all know that current approaches, they rely on correlative species distributions and really, really coarse scale climatic data for the marine environment as well. We don't have very, very good, climate data at all. So we're now starting to move beyond correlative species distribution models, and especially with novel climates, we need to have something that's going to be a little bit more accurate. And this involves niche tracking in multivariate space. So we don't just need to know where the organisms are distributed, we also need to have a more mechanistic understanding of what these species of invertebrates and macroalgae do. We need to have quantitative data on physiological performance, their environmental sensitivities, how they acclimate or adapt, and biological interactions as well, because no single species lives in isolation. So the way that we do this is to try and bring all of these factors together using an eco-mechanistics approach. And this is just a basic schematic of what my research group does. We use microclimate data, which is up on the top left there. We then also do in situ measurements of body temperature. There's a robo-limpet second there. We bring animals into the lab and we do long-term mesocosm experiments and look at physiological parameters as well. We're now looking at thermal imaging, so this is a lovely thermal image of the intertidal zone there as well. We bring all of this information together from these experimental work with the long-term time series. Unfortunately, nobody in the marine environment has actually drawn a species distribution model that looks like anything marine. So I've had to borrow Mike Kearney's lizard model just so you can see the way that the parameters are pulled together in that model. But hopefully this gives you a little overview of how we're now trying to make biological sense of a changing world. We're not just trying to correlate differences in coarse scale climate and ocean acidification with large scale biogeographic changes. We actually want to know what's actually happening inside individual organisms and how is that spreading out into populations, communities, and then biogeography as well. We do this by collecting time series, like the Markram time series. This is a graph here of barnacles that we have that goes back to 1952, and it's been collected every single year since then, so it's a fantastic resource. We've also got experiments out on the intertidal, again for barnacles here, so we're looking at different competition and settlement and recruitment rates, and there's these two right pictures here. So we go out and do a huge amount of field work and field experiments. Because we're interested in putting biological realism into the models, we're also looking at biological responses as well. And so this is again why we don't use any means of large-scale climate data, because as you can see here from the left graph, this shows that if you shift the mean temperature ever so slightly with those arrows, you actually change the amount of cold and hot days completely disproportionately. So we need an awful lot more resolution in our environmental data as well as the biological data as well. So we're testing the response mechanisms. The Marine Biological Association has a really good mesocosm system up here where we can alter all sorts of different parameters: light, temperature, tidal range, salinity, pH, nutrients, microplastics. We combine that with physiological work, and then of course, because I just love fieldwork, we get out and do a huge amount of fieldwork as well, all across the distributional range of species. And then we try and bring all this together with these mechanistic species distribution models to finally actually get some realistic information about where species are and where they're going to move to in the future. I'm just going to quickly run through one example of what we're doing here. The blue mussel, Mytilus, actually has a hybrid that's around the whole of Europe, and this is a hybrid between Edulus and Gala provincialis. And quite conveniently for me, because I'm based in Plymouth, the hybrid zone covers the whole of the southwest where we are. So I brought hybrids in because people have done some work on one species or another species, but because hybrids are present across Europe, we've no idea what happens with them. And we set our multiple stress and mesocosm up, so we had different temperatures, different pHs, and then we also added microplastics as well, just to see how all these different stresses in combination would affect them. And one of the results I was really surprised about is we actually saw a change in fundamental body size scaling in some of the treatments. The ones that had higher temperature increased carbon dioxide, so a reduction in pH, and plastics. They actually changed from this universal scaling rule, and we've never seen anything like that before. So this was obviously quite a stark response to these multiple stresses that we really have to start working on a lot harder. And some of the other impacts, well, the addition of all these different stresses increased mortality, which is really bad thing. Oh, and also I do my presentations on a Mac, so apologies that these are coming up out of order. But also the addition of microplastics increased the body weight. Now, microplastics that we were using are so small that they weigh nothing. But they are fundamentally changing the body weight of these animals. The effects were more severe at higher CO2 levels, so as the pH decreased. The length of the shell increased under warmer conditions as well. And we keep seeing this. The effects of everything are more severe at higher carbon dioxide concentrations. So ocean acidification on top of climate change is really starting to stress these species out. And then if we start to put man-made pollutants in the water as well, unfortunately, a lot of these species are really going to suffer. This is just one example of the species distribution maps that we do for species, and this one just shows you the Mediterranean, because I thought, well, I've depressed you all now for 10 minutes, I may as well take you somewhere a little bit nicer and warmer than Liverpool, because I have to say it's a bit cold here. And this just shows you now how we can produce much more realistic maps of where the current species distribution is, but as soon as we start to add different factors in there as well, so when we add salinity and eutrophication, you can see that the models change, and the one on the bottom right is the one with the multiple stresses. And it can now pick out really small areas of coastline that are going to be still within the biogeographic range, so not at range edges, but are really going to start to struggle in the future as it gets warmer, as the salinity changes, and with eutrophication, so that managers can actually have practical tools to say, well, we shouldn't put a farm here, or we shouldn't do this here, and we're going to really struggle with conservation here. We need to take these additional factors into account. So finally, we're actually starting to get like spatially relevant, really good species models. This is just a very quick schematic, basically, of the reason that I don't have any life. Because, you know, we're looking at global and regional scale problems, but what we need to do then is downscale to look at physiological properties, how these scale up to the organism, and then when we build these models back up, eventually we can make really good predictions at the ecosystem level. So hopefully I've given you enough information to slightly, you know, tease your interest. I'd just like to finish by saying, I love all my travels around the world to look at where things are, and thanks to all these people, I keep getting funded to go on holiday. Sorry, really nice research trips. So thank you.
So a very comprehensive integrated talk. It must generate some questions. Too integrated, too comprehensive.
That's all right. That's all right. We've got a website. People can go and look at that.
That very last bit, when you said we've got these tools now that managers can begin to use.
Yeah.
How on earth do you get them to use them?
Well, we've got a very strong science policy link, so I'm a member of several like policy relevant groups like the UK, the UCMAS group and HEBSEG, which is the Healthy and Biological Diverse Seas Evidence Group for the government. The Markland project is funded by Natural England, Natural Resources Wales, Scottish National Heritage. So we have direct links with these people that actually want to fund this kind of data collection and development to answer an awful lot of the deliverables that they're charged with doing. So thankfully, at least for the coastal marine environment, we have direct access to people that can make decisions and then will tell us, well, can you produce your results at this scale for this project or this, you know, looking at SACs, MCZs, if they'll still exist, and all that relevant size conservation and management work. They'll come to us and say, can you produce information at this spatial or temporal scale? And thankfully now we're just about starting to get to almost like a bespoke on-demand service.
That's brilliant. So if you're sitting on the island of Cyprus and you have your conservation-minded people, they will be able to communicate with the planning?
Hopefully, yes.
Thank you very much, Nova. We must move on now. The speaker is Thomas Nova. Thomas Davies, and he's talking about Ecology, the impacts of artificial nighttime lighting on temperate grass and
Okay, so Nova's talk highlighted a variety of different ways in which mankind is affecting ecosystems. But there's one which we've missed for a long time, and that is artificial light pollution. This is a recent satellite image taken in 2012 via the Suomi sensor, and it shows the world at night. This year, Fabio Falchi and colleagues published a paper in Scientific Advances, the second World Atlas of Nighttime Sky Brightness, where they took one of these images and they combined it with an atmospheric dispersion model to give us an impression of how bright the night sky is from an observer on the ground. And they estimated that 23% of the world's land surface, cut roughly to where this map is here, is artificially lit at night. So it's clearly globally widespread. And I just want to make 2 points about artificial light in comparison to other global change stressors. The first is that large fluctuations in carbon dioxide concentrations and global temperature have been experienced by ecosystems throughout evolutionary history. Changes to natural light regimes are completely unprecedented. And the second is that even if we stopped producing CO2 emissions tomorrow, it would take centuries for carbon dioxide concentrations in the atmosphere to reach their pre-industrial levels. We can turn the lights off now. So this is a really virgin subject area. It's only been going since around 2004, and in the last 6 years, it's really become consolidated into its own research field. And most of these taxa have now had impacts demonstrated, impacts of artificial light demonstrated on various aspects of their biology in the last 6 years. And the number of papers demonstrating the ecological impacts is growing exponentially currently. On the Ecolight project, we're primarily interested in ground-dwelling invertebrates. Plant Life International recently estimated that 238,000 hectares of road verge present in Britain. That's roughly twice the area of semi-natural grassland. And they are also the principal habitat which are lit by street lighting. These road verges are really important habitats for pollinators. They're also really important as corridors for the dispersal of ground-dwelling invertebrates. So when we started the Ecolight project, we did a quick look-see study in 2011 where I went out to a row of streetlights outside the town of Helston and put out pitfall traps directly underneath the streetlights and between the streetlights. And we trapped for a very short period, 3 days and 3 nights. And we separated our traps by day and night. And what we found was really interesting. Firstly, all of these groups were aggregating around the lights. So the traps directly underneath the lights are the open bars here, and the traps in between the lights are the grey bars. The second really important thing that we found was that they aggregated around the lights regardless of whether it was day or night. So they weren't simply aggregating at night and then redispersing during the day as you might expect for Lepidoptera. The lights were having a fundamental effect on the spatial distribution of the organisms across that roadside verge that was permanent in time. If you switch the lights off, they might recover relatively quickly, but because the lights come on every single night, that effect persists. And street lighting is changing, and it's changing rapidly. When we started the Ecolight project back in 2011, light-emitting diodes represented 9% of the lighting market. We knew by the time we finished they would be big. They're 45% of the lighting market. By 2020, they'll be 69% of the lighting market. Lighting is changing, and it's changing fast. And as far as organisms are concerned, these changes are really important. This figure here compares 2 spectral power distributions. One is for an LED lamp, it's the double-humped curve, and one is for an old low-pressure sodium light of the type that we used to use commonly in the UK back in the 1960s. That's this orange curve. The scale is between 0 and 1. It's standardized so that you can compare the different spectral power distributions. What you can see is that LEDs illuminate across a whole range of different wavelengths. And by comparing them to the range of wavelengths that insects or arachnids can detect light, you can see that the likelihood is that LEDs will facilitate color-guided vision at night a lot better than LPS lights. So that's the first problem with LEDs, but there's 2 others. The second is that the majority of biological responses are wavelength specific. And those responses occur right across the spectrum. So by using LEDs, you increase the probability that you will cause some type of biological effect. The third is that the majority of known animal responses to artificial light occur in the short wavelengths, around 350 to 500 nanometers. And you can see from looking at the spectral power distribution that LEDs have a strong peak in the blue where they're emitting a lot of light. LEDs are also part of the solution, though. The technology is very flexible. The lights can be dimmed, the spectra can potentially be manipulated to eradicate this blue peak, and they can be switched off at night. And in the wake of the financial crisis, local authorities across the UK have been taking advantage of LED lighting technology by introducing LEDs and then switching them off at night in order to save energy. Currently, there are 23% of local authorities have permanent nighttime switch-off schemes where they switch off the lights between midnight and 4 a.m. And 39% are engaged in dimming schemes on a permanent basis. So on the Ecolite project, we were really interested in comparing those different mitigation strategies in two sister experiments. In the first, we have a field experiment where we illuminate a previously light-naive landscape and monitor the changes in invertebrate composition over time. And in the second, we are comparing the different lights against different levels of trophic complexity so we can elucidate something about how the light affects trophic interactions. So to start with the mesocosm experiment, we have a fully crossed design in which we have an amber LED, which is a very close approximation of an LPS light, a white LED of the type used in street lighting, and controls, an unlit control. And that's fully crossed with 3 levels of trophic complexity: plants only, plants and herbivores, and plants, herbivores, and predators. And within all of those trophic groups, we have both a diurnal and a nocturnal predator. And what we find in that experiment is, firstly, that the number of flowers produced by species of lotus is reduced underneath amber light, and that suppresses the aphid populations. The second thing that we find is that for those mesocosms that had carabids in, the slug populations were significantly suppressed under the white light. So both of those different types of light cause trophic effects. In the field experiment, we compared our white LEDs to the amber treatment again, but we also compared to a dim treatment and a treatment that was both dimmed and switched off at night. And these were distributed around our experimental site, as you can see here, and there are controls interspersed amongst that landscape. And we're also, in each instance, we're comparing the impact of our different strategy by comparing it to our controls. And you can see here, these are the different lux levels of our lights. Lux is a measure of brightness to the human eye. It's not particularly biologically relevant in an ecological context. However, it's an industry standard. And if we were to try and present a measure that was ecologically relevant, we could only measure it as lux relevant to the organism of interest. So our dim treatments are roughly 50% brightness in comparison to our high-intensity white LEDs. And then we have our ambers, which are slightly above that. And what we find is that in terms of the number of taxa affected when compared to the controls, the high-intensity white LEDs clearly have the biggest impact. They impact 7 taxa within our system. Most notably, the large carabid Pterosticus niger and the large nocturnal lycosid Trichosa ruricola. Again, reflecting back on our mesocosm experiment, there's clear potential there for top-down effects. Also, a whole range of different linephid species are affected. 1 tetragnathed, Pachygnatha jagiri, and some other coleoptera. So 7 species were affected by the high-intensity whites, 4 by the ambers, 4 by the dimmed whites, and 2 by the dim white timers. So it seems like a combination of dimming and switching the lights off in the middle of the night is the best mitigation strategy for getting around the impacts of artificial light on grassland invertebrates. The graph on the right-hand side shows us the same comparison, but this time broken up through time. What's interesting about our site is that we had a change in species composition through time, which we think at the moment was caused primarily by the fact that we erected a fence around our plots to prevent the sheep from getting in and grazing them. And as a result, we changed the management regime in that area. And so we see the assemblage gradually recovering from one that's dominated by linephid spiders to one that has more beetles in it. And what's important about that is that we see these effects at different times. So the linnefield effects we saw early on in 2012, but the Caribid effects we saw later on in 2014. So this suggests that when you're lighting up a roadside verge, you're not just having an impact on the organisms within the roadside verge, which largely the community composition of that type of habitat would reflect a better managed system. Whereas in 2012, our community composition reflects maybe more of an agricultural system. So we're not just having an effect on the roadside verge, we're probably also changing the invertebrate composition within the adjacent field as well. So just to wrap up quickly, I just want to state that artificial light at night is a globally widespread anthropogenic stressor. It has ecological impacts that are seen in grassland ecosystems. These impacts are seen both with the old and the new technologies. So reverting back to old-style technologies doesn't seem to be a solution. But what does seem to be a solution is switching the lights off in the middle of the night at the same time as dimming them down. Thank you.
Do you think you would find similar issues in woodlands, for example? I mean, many streets are tree-lined, and that you'd get marked switches under different light regimes?
Potentially in woodlands, yes. Woodlands present an interesting case because the vegetation itself acts as a barrier to light. So whilst you would expect to see similar effects, you'd expect them to be more localized, because the distance which the light spreads, and so the distance over which an organism can detect light, is reduced.
Any other questions? Yes, Mike.
Have you had any conversations with local authorities about acting on this?
Yes, we have. We've had conversations with our own local authority in Cornwall. However, at the time we had those conversations, they'd recently just gone through a program of replacing all their lights with metal halide lights, which actually emit somewhat in the UV and are even worse than LEDs. And they didn't like what we had to say. I've been in conversations with lots of different lighting engineers up and down the country, and there seems to be a general consensus amongst them that they are very keen to balance up the benefits of light with also reducing ecological impacts. So, I mean, it's clear to me that LEDs present as much of a solution as a problem. The question is whether or not the lighting industry wants to get on board with that. I was recently at a conference where a prominent member of the lighting industry who worked for a particular company who makes a lot of light bulbs stood up and effectively said that whilst they recognized that they could produce more environmentally friendly lighting technologies, there simply wasn't the market for it. So it's the lighting suppliers clearly aren't accepting any responsibility from their end. They want to see the consumer demanding more ecologically friendly lighting first.
Okay, well, we must move on to our last speaker now. This is Sean and he's talking on tree vital rates as vital traits in understanding forest dynamics under climate change.
Right, thank you very much, everyone. Is there a laser? Okay, I'll go without a laser. So one of the important challenges in ecology and evolutionary biology now is understanding how whole systems will respond to environmental change. And this is particularly because we know the answer is complex, because whole systems are made up of species. Species respond differentially to environmental change. We know that because that's one reason why they're species. And so moving forward, we can't model all species, and we need to think about how to aggregate species. That is, collect species in a meaningful way that simplifies the problem but retains some of the key elements and ways in which performance is affected by the environment. I say moving forward, but this has been a problem for any system or any project that tries to do kind of holistic modeling. And Earth systems models and terrestrial biome models in particular are an example of that. So basically, a terrestrial biome model, which is trying to understand global changes in vegetation, has to use something called plant functional types, that is, groupings of species that share similar parameters or rule sets. So these are the plant functional types for the community land model, CLM. And this is for all of Earth's forests, basically, and they're divided based on whether you're in the boreal zone, temperate zone, or tropical zone. You have needle leaves or broadleaves, or you're evergreen and deciduous. And it's important to note that the most diverse terrestrial biomes, tropical forests, are represented by two functional groups. Now, the people that work on this know that this is a problem. And one of the ways they're thinking of making these groups more mechanistic and maybe more physiologically based is by looking into aggregating species according to functional traits, particularly traits that are commonly measured and might indicate some kind of strategy, especially strategies with respect to environmental change. Essentially, the way this works is that a trait will confer some fitness advantage, say, given a certain environment. So if you have a trait that confers drought tolerance, there's a drought, well, then that will give you an advantage versus not having that trait, clearly. There's some problems with this, and I want to move through the problems and talk about a way to kind of reset this approach, basically, to aggregating species. One of the problems is that when you start to scratch the surface, you find that there's a model of very equivalent traits and infinite environments, and so you can kind of have your favorite trait environment interaction, and you sort of end up where you started with a system that has so many components, potential components, that you're kind of back at just modeling a high-dimensional problem. Instead of species, you have all these different ways of matching traits to an environment. Another problem is that there's often a mismatch between the kind of presumed role that a trait will have in a life history strategy and what it actually might do. An example here is wood density. Wood density is thought to be a good indicator of where you fall in the fast-slow continuum. That is, dense wooded species tend to survive. They think of it as an investment in survival over growth, and vice versa. But, for example, work by Osazua Peters and others have shown that in tropical trees, for example, you can have the same density of wood, but have tissues that either confer growth or survival advantages. In other words, you can be a fast-growing species with dense wood. And so disentangling exactly what the actual role of the trait may be difficult. Another problem is in the data problem. So specific leaf area, for example, this is specific leaf area on the x-axis and height on the y for temperate forests, is thought to also indicate strategies, demographic strategies. But here you see where you get your data from and how you decide what a species' average trait is can be problematic. In fact, there's a lot of work now with this kind of, these kinds of traits showing that interspecific differences are really important in this. So for example, if you sample from the top of the tree, all the species are the same. If you move down, where you sample really matters. And so this can also be problematic when you're trying to aggregate across diverse systems. Another thing is the lack of, I see a lack of evolutionary biology in some of these problems. And an example of this is that, well, an evolutionary goal of organisms is to create something that can create something. In other words, reproduce and confer an advantage on your offspring to reach reproductive stages. So here you have survival on the y-axis, and on the x-axis is size. So you can think of that as ontogeny. And so you may have a strategy where you invest in high survival through lifetime, you reach reproduction, which is the dotted line, and you keep investing in high survival. So seed output starts there, and you senesce slowly. We also know, though, that this can change radically. Once you reach reproduction, you can have a completely orthogonal strategy. In fact, you can slowly grow, survive high to reproduction, and then immediately reproduce and die. And this is monocarpic strategy, and there are trees that are monocarps that take decades to reach the canopy, flower, and die. So appreciating the difference between how you invest in a strategy before and after reproduction may also be important. Oops. And finally, I think that there's, at least in terms of the data we receive, it's unclear exactly what's related to an allocation strategy and what might be due to acquisition. And again, I'll use wood density. Wood density is thought to be a way of allocating resources to a particular life history strategy, say high survival. But also, if you lack resources, that is, you're not acquiring carbon, you may grow very slowly and end up with very dense wood. In other words, you can have dense wood because you're suffering and you're suppressed, or because that is a life history strategy, and it's hard to disentangle that. So what's my proposed answer? First, it's not to ignore or get rid of the idea of traits as being important components to, important tool to use in this type of problem. And nor is it to ignore all the important complexities of the environment by trait relationships. But it's to sort of reset the system in terms of vital rates again. In other words, for the time being, let's set aside traits and focus more on the survival, growth, and reproduction and what that means. And instead of thinking about that in terms of all the myriad environmental contexts, let's think about it just in terms of time and space. So simplify by just saying, how are you growing and surviving given where you are and when you are? And then we can later add in some of these environmental variables and complexity and some of the trait, unique trait qualities. So essentially, it's, the goal is to, I was hoping to have this here, estimate vital rates in a way that appreciate life history strategies, but also in terms in the context of allocation and acquisition. So to do this, in this example especially, we're modeling survival and growth. And the survival you can see here, we're fitting a form that can appreciate some of these ontogenetic changes, mechanistic differences between how you survive when you're small, through thinning, your maximum survival rate, senescence, different life forms, and also changes over time. And growth, and I'm sorry, so this is we fit two logistic functions to this, and the parameters kind of map to these parts of life history strategies. For growth, we basically fit how you grow when you're suppressed, that is, you're small and don't have access to resources, versus how you grow when you have access to resources, you're small and you're in a gap, say, so the top 5% of growers, or if you're canopy or reproductive. So it's assumed that the fast growers have access to resources, and it's a simple response. X-axis is just millimeters of growth, and these distributions are fit to the species. So the data that I'm going to show you here are from BCI and Panama, and essentially we fit Bayesian models of those functional forms, the gammas and the logistic functions. And so you have 3 censuses for all these species, 120 species in total, and all of the posterior estimates of these functions, of these functional forms of the parameters for them. And we can take all those parameters and throw them in a PCA and find out where these parameters fall in this kind of higher dimensional space. And we do see that, for example, that elements that fall out, this is just the first 2 principal components, for example, demonstrate some of these key strategies I'm talking about. Over-ontogeny, so the survival of small and large individuals, overall survival rates over the long period, and growth rates. And they fall out in unique ways. So we can throw all of the species and censuses back at this PCA and cluster them in high dimensions again and see what comes out of this. So we have 8 clusters. This is an example of 5, but we have 8 clusters that I'm going to show you. And essentially what we're looking for is ways in which the species mass is partitioning this life history strategy space. And so I don't have time to go into details, but you can clearly see the life forms fall out. The x-axis is size in millimeters, and the y-axis is survival. And of course, and for the small squares, the x-axis is growth, and it's just a distribution. So essentially, if you look into it, you can see slow, fast growth. You can see some interesting, potentially important things like, for example, here where this looks like a little, it wouldn't matter much, but this can indicate whether or not a cohort of small individuals is actually getting into the population. And you'll see differences across that. But let's bring it back to the traits we were talking about. This is trait data from Joe Wright's work at BCI. And so here are the 8 clusters, and we have maximum height. And you see that, yes, maximum height falls out with our life history forms. Seed mass, there's not much there. LMA and wood density actually show very little relationship except for that first group, perhaps. And if you zoom in on that, these are the understory, the kind of the smaller individuals. That low wood density, or so the dense wood here, looks to be more of an acquisition issue. In other words, this is a low surviving, slow growing cluster. And so in other words, these are species that are suffering, and they show low wood density. Otherwise, it's kind of all over the shop. So at least in this pass through these data, you're really not seeing these species traits map to the life history strategies of the way they're supposed to. Further, we can kind of just project this forward, and you can just look at, this is, sorry, I lost the x-axis, that's just years of simulation. So these are simulated years, and you see a cohort of 10,000 individuals for each cluster kind of moving through the space. And so you can see how some clusters represent fast growth canopy level, slow growth canopy level, different levels of carbon acquisition, different levels of carbon retention, et cetera. So you can kind of see how the community falls apart, this falls into these really, really unique clusters in life history strategy space. So future work is to apply this paradigm to global data set, and Jessie Needham, a postdoc in my lab, who's just starting, she's now collecting data sets from all over the Forest Geo network. And so we're really looking to try to build like a kind of a worldwide demographic economic spectrum. In other words, how do all of these species fall on these axes? And where do you see things like cyclone forest or forest across a seasonal gradient, et cetera, fall out? Further, we want to revisit some of the classic trait-based analyses, looking at how these clusters might be phylogenetically conserved within communities, looking at spatial patterns, et cetera. So there's a lot to be done in the same way. And I should add also, I want to map traits back on to these clusters. In other words, which physiological traits, leaf venation, hydraulic traits, might actually be really good indicators of the life history strategies we see fall out. And then finally, working with people at Berkeley Labs and NG Tropics, looking to use these clusters as ways to benchmark and guide terrestrial biome models. In other words, are your models capturing the slow turnover time versus fast turnover time that we see in these different clusters given environmental variables? So with that, thank these folks and take any questions, please.
Thank you. So if you found that some of these traits correlate really well with these clusters that you're picking out, would you go back a step and say, okay, now I've used this process to find the traits that are actually meaningful and use those instead? Or do you think the clustering?
Yes, I mean, the demographic data are great. That's a great question. The demographic data are great, but yes, there is an advantage to having the traits, because if you can collect them easily, you can go into a forest and snip leaves or do microcores. And yes, that would be an excellent short. So ideally, we would find the traits and say, target these species in this forest if you want to understand these groups. So, yeah.
Any other questions? Just very quickly, you've obviously worked in Panama, but do you think in more temperate forests that you would find a similar difference between fast species and slow species and the traits?
I would expect, I do work in temperate forests also, and in fact, that's one of the motivations for this, is I think that just like traits where you see low diverse systems having a wide range of traits, I would expect the same thing in temperate forests. We have fast-growing, slow-growing individuals, here in the UK, and that's one thing we want to explore, is exactly how common that is, that you can map a very simple system, a boreal system, and it completes the life history trait space that a tropical system might.
Okay, well, thank you very much indeed.
All right, thank you.